Hi class, this is your instructor, Skylar Huff. We're now in Chapter 27. This is the prokaryotes. All cells can be characterized as one of two fundamental cell types, prokaryote or eukaryote. The term prokaryote describes a cell type or, collectively, the organisms in the domains bacteria and archaea. Although all replicating cells share the common characteristics of having ribosomes, cytoplasm, a plasma membrane, and DNA as their genetic material, there are important differences between eukaryotic and prokaryotic cells. Prokaryotic cells are significantly more abundant than eukaryotic cells. Estimates suggests approximately 10 to the 31st power prokaryotic cells, and 4 to 5 orders of magnitude fewer eukaryotic cells in the biosphere. In the human body, there are about the same number of bacterial cells as there are eukaryotic cells, but the balance can vary to favor one or the other cell type with just one bowel movement. Although some prokaryotes are important agents of potentially deadly diseases, like bacteria, Pseudomonas, or Goinsena, pictured here, an overwhelmingly greater number play essential roles in nutrient cycling and energy transformations in ecosystems. Without these prokaryotes, life as we know it may not have appeared, and most definitely could not continue. While the absolute number of prokaryote species remains unclear, it is likely that there are many more species of bacteria and archaea than there are species of eukarya. Still, difficulties of defining species aside, it is not the number of species of prokaryotes versus eukaryotes that is interesting. More important is the diversity of ways prokaryotes are successful relative to eukaryotes. Prokaryotes colonize environments that would be lethal to most eukaryotic organisms. The metabolic and physiologic diversity of prokaryotes has allowed their success in virtually all ecological niches ever explored. Because prokaryotic cells are generally significantly smaller than eukaryotic cells, they remain hidden until the mid-17th century when Anton van Leeuwenhoek observed what we now know to be bacteria using a homemade microscope. Even before this, however, Girolamo Fracastoro suggested that disease was caused by unseen organisms. The history of microbiology entwines these two strands of improved technology for visualizing the unseen, microscopy, and investigating the nature of infectious diseases. Central to this was also the prolonged debate about the very nature of how living things arise from other living things, or by spontaneous generation from non-living matter. The controversy over spontaneous generation continued into the 19th century when Louis Pasteur and others conclusively shown that life does not arise spontaneously. Ultimately, Robert Koch was able to identify and culture the causative agent for the disease anthrax. He proposed the following 4 postulates, Koch's postulates, to prove a causal relationship between a microorganism and a disease. 1. The microorganism must be present in every case of the disease and absent from healthy individuals. 2. The putative causative agent must be isolated and grown in pure culture. 3. The same disease must result when the cultured microorganism is used to infect a healthy host. And 4. The same microorganism must be isolated again from the diseased host. Even Koch realized that these postulates do not hold true for every case of infectious disease, however. For example, many people infected with the bacterium causing cholera are asymptomatic, and therefore the first postulate is violated. A causal relationship between an infectious agent and a disease is better established using a set of molecular criteria, some of which bear resemblance to Koch's original postulates. The study of microorganisms and disease also led to advances in knowledge about the immune system and how exposure to a disease-causing agent can prevent future disease. Due to our increasing knowledge about the role of prokaryotes in ecosystems and how they can be manipulated to produce useful products, microbiology is more relevant than ever. We can now survey microbial diversity using an approach called metagenomics. DNA is sampled directly from an environment and sequenced. Then, using computer algorithms to assemble genome sequences, new species can be identified. This has revealed even greater diversity of prokaryotic forms than suspected, along with novel adaptations to diverse environments. Although we recognize thousands of different kinds of prokaryotes, it is estimated that less than 10% of prokaryotic species have been identified and or characterized. Prokaryotes are the oldest and structurally simplest and likely most abundant forms of life. Prokaryotes were abundant for over 1 billion years before eukaryotes appeared. Early photosynthetic bacteria altered the Earth's atmosphere by producing oxygen, which stimulated the expansion of prokaryotes into oxygenic environments and facilitated the appearance of eukaryotes capable of utilizing oxygen. Prokaryotes are ubiquitous and live everywhere eukaryotes do, but also flourish in places no eukaryote could live. Bacteria and archaea have been found in deep-sea hydrothermal vents, at volcanic rims, and deep within glaciers. Some of the extreme environments in which prokaryotes can be found would be lethal to any other life form. Many archaea are extremophiles. They live in hot springs, in hypersaline environments that would dehydrate other cells, and in atmospheres rich in toxic gases such as hydrogen sulfide. Some of these harsh environments may be similar to the conditions present on Earth when life first appeared. It is possible that prokaryotes have adapted to survive these harsh conditions early on and have retained the ability to exploit these areas as the rest of the atmosphere has changed. Prokaryotes differ from eukaryotes in many important ways. These differences represent some of the most fundamental distinctions that separate any groups of organisms. They are as follows. Unicellularity. With few exceptions, prokaryotes are single-celled. In some species, individual cells adhere to one another and form filaments or aggregates. However, the cells retain their individuality. Even in these cases, the characteristics of the organism are the characteristics of the individual cells, unlike in a multicellular organism. In their natural environments, many prokaryotes appear to be capable of forming complex communities of different species called biofilms. These associations are more resistant to antibiotics, desiccation, and other environmental stressors than is a simple colony of a single type of microbe, such as might grow on an agar plate in the lab. Cell size. As new species of prokaryotes are observed, investigators are finding that the size of prokaryotic cells varies by as much as 100,000-fold. Thiomargarita namibiensis is the largest known bacteria with cells up to 0.75 millimeters in diameter, just visible to the naked eye, and is shown in figure 27.3. Most prokaryotic cells, however, are 1 micrometer or less in diameter, whereas most eukaryotic cells are about 10 times larger. While this is a useful generalization, there are very small eukaryotic cells as well as very large prokaryotes. The nucleoid. Prokaryotes lack a membrane-bound nucleus. Instead, they usually have a single circular chromosome made up of DNA and histone-like proteins in a nucleoid region of the cytoplasm. The exceptions to this are Vibrio cholerae, which has 2 circular chromosomes, and Borrelia species, which have multiple linear chromosomes. Prokaryotic cells often have small circular DNA molecules called plasmids, which usually confer some selective advantage but are not essential. cell division and genetic recombination. Cell reproduction in eukaryotes takes place by mitotic or meiotic cell division and uses spindles made up of microtubules to segregate chromosomes. Cell division in prokaryotes takes place mainly by binary fission, which does not use a spindle. Prokaryotes also do not have a sexual cycle, although they do exchange genetic material extensively. These mechanisms are collectively called horizontal gene transfer. While not related to reproduction, they play important roles in evolutionary change. Here, this is showing that biofilm, and this is found in the dental plaque on a toothbrush bristle. Brush your teeth. Internal compartmentalization. Prokaryotes do not have extensive membrane-bound organelles like eukaryotes. However, some species have structures that can isolate specific metabolic pathways or provide storage. These structures may be bounded by a lipid bilayer or monolayer, or by a protein shell. The plasma membrane can also be extensively invaginated to isolate processes like cellular respiration or photosynthesis. Flagella. Prokaryotic flagella are composed of a single protein fiber and not that 9+2 arrangement of microtubules found in eukaryotic flagella. Bacteria flagella have a twist, are rigid, and rotate to drive cell movement. Eukaryotic flagella, on the other hand, have a whip-like motion. Pili. Some species of bacteria have numerous filaments of protein extending from their surface called pili or fimbriae. These are structurally diverse and may be found to be involved in the exchange of genetic material, in the attachment of pathogenic species to host tissues, and occasionally in motility. Metabolic diversity. The metabolic diversity of prokaryotes reflects the diversity of environments in which they can live. Different prokaryotes can exploit a variety of organic and inorganic electron donors and acceptors to perform cellular respirations, can ferment a variety of organic substances, and use a variety of different carbon sources. In these respects, there is significantly greater metabolic and nutritional diversity among the prokarya than amongst eukarya. Some bacteria are photosynthetic and may produce oxygen, and some other oxidized byproduct. Archaea and bacteria are similar in that both have prokaryotic cellular structure and are ancient forms of life. However, they vary considerably at the biochemical and molecular levels. Moreover, phylogenies based on rRNA sequences show that the archaea are more closely related to eukarya than they are to the bacteria. Excuse me. They differ in 4 key ways: the plasma membrane structure, the cell wall structure, DNA replication, and gene expression. There are also some metabolic differences between the 2 domains. The plasma membrane. All prokaryotes have it. The membranes of archaea differ from bacteria and eukaryotes. For instance, archaea membrane lipids are composed of glycerol linked to hydrocarbon chains by ether linkages, not the ester linkages seen in bacteria and eukaryotes. And the hydrocarbons, they may be branched. The cell wall. All prokaryotes have a cell wall. Bacteria have peptidoglycan. However, archaea lack peptidoglycan. With the DNA replication, both have single replication origin, and the nature of origin and proteins used are different. And then archaeal DNA replication is more similar to that of eukaryotes. And finally, gene expression. Archaeal transcription and translation are more similar to those of eukaryotes. So here we have it. So we have the common ancestor of life on Earth, and then you have firstly domain bacteria, then there is domain archaea, and then lastly domain eukarya. Prokaryotes are not easily classified according to their forms and only recently has enough been learned about their biochemical and metabolic characteristics to develop a satisfactory overall classification scheme comparable to that used for other organisms. With no clear definition of what constitutes a prokaryotic species, systematics has been difficult. Early taxonomies for classifying prokaryotes relied heavily on observable or microscopically detectable differences between organisms or groups of organisms. Key characteristics initially used to classify prokaryotes were photosynthetic ability, cell wall structure, motility, unicellular or colony forming or filamentous, spore forming ability, and pathogenicity. As technologies have advanced, combinations of approaches have been used to create taxonomies. For example, information from genomics can be combined with phenotypic and phylogenetic data to create more meaningful and well-informed classifications. Molecular approaches include the following: the analysis of amino acid sequences of key proteins, the analysis of nucleic acid base sequences by establishing the percent of guanine and cytosine, nucleic acid hybridization, which is essentially the mixing of single-stranded DNA from two species and determining the amount of base pairing. Fourthly, gene and RNA sequencing, especially looking at ribosomal DNA sequences. And lastly, whole genome sequencing. So as I showed just a moment ago, the three-domain system of phylogeny, originated by Carl Weiss, relies on all of these molecular methods, but emphasizes the comparison of ribosomal DNA sequences to establish the evolutionary relatedness of all organisms. That rDNA sequences were chosen for their high degree of evolutionary conservation to ask questions about these most ancient splits in the tree of life. Based on these sorts of molecular data, several groupings of prokaryotes have been proposed. The most widely accepted is that presented in Burgess' Manual of Systematic Bacteriology, 2nd edition, most recently published in 2012. At the same time, large-scale sequencing of randomly sampled selections of bacteria shows an incredible amount of diversity. These new data indicate that the vast majority of bacteria have never been cultured and studied in any detail. So you're seeing here figure 27.6 with some major clades of prokaryotes. And this is from Burgess' Manual of Systematic Bacteriology. So you have Archaeota, Aquafaceae, Bacilliae, Actinobacteria, the spirochetes, the cyanobacteria, beta, gamma, and delta, and the rest is shown here. Prokaryotic cells are relatively simple, but they can be categorized based on morphology. They also have some structural variations that allow them to be differentiated with stains. Although it is an oversimplification, it is useful to classify bacteria based on obvious morphologies. Most prokaryotes exhibit one of three basic shapes: rod-shaped, often called bacillus; coccus, also called spherical or oval-shaped; and spirillum, long and helix-shaped. Spirochetes appear similar to spirilla. However, they tend to be narrower with a looser twist. Some types of prokaryotes have no specific shape and are said to be pleomorphic. The bacterial cell wall is the single most important contributor to the cell shape. Bacteria that normally lack cell walls, such as the mycoplasmas, do not have a set shape. Some rod-shaped and spherical bacteria form colonies adhering end to end after they have divided, forming chains. Some bacterial cells change into stalked structures or grow long branched filaments. The range of cell morphologies seen in prokaryotes make it difficult to describe cell size. However, a reasonable size ranges from 200 nanometers to 0.7 millimeters. The average bacillus is anywhere from 0.5 micrometers to 3 micrometers in diameter and up to 15 micrometers long. For comparison, the average eukaryotic cell is about 20 to 25 micrometers in diameter, but again, there are significant variations. More important than absolute size ranges are the consequences of being small. The coccozoid-shaped bacteria, with a cell diameter of 2 micrometers, has a surface area to volume ratio of 3. As the cell diameter increases, the surface area to volume ratio drops rapidly. A simple doubling of the diameter results in the halving of the surface area to volume ratio. This ratio is important because nutrients are taken up across the surface of cells. The more surface area a cell has in proportion to the amount of volume, the more rapidly it can take. For a cell to divide, the cell must grow to a certain size and replicate its chromosome or chromosomes, processes requiring nutrient uptake. Prokaryotic cells that are smaller than eukaryotic cells can rapidly absorb nutrients, grow rapidly, and quickly replicate their chromosomes. Every time a chromosome replicates, there is a chance for mutations. A rapidly dividing prokaryote can accumulate mutations that it can test in its environment for a selective advantage. Subsequently, small cells evolve more quickly than do larger brethren. This is particularly problematic when bacteria can test out mutations in a few generations to evade antibiotics that take us years to develop and test. Most but not all prokaryotes possess a cell wall and some number of other external structures. A cell wall, if present, is often complex, consisting of many layers. Minimally, it consists of peptidoglycan, a polymer composed of a rigid network of polysaccharide strands cross-linked by peptide side chains. The cell wall maintains the shape of the cell and protects the cell from swelling or rupturing in hypotonic solutions, which are commonly found in the environment. The archaea do not possess peptidoglycan, but some have a similar structure called pseudomurin. In bacteria, the structure of the cell wall can be used as the basis for creating two groups, the gram-positive and the gram-negative bacteria. Two types of bacteria can be identified using a staining process called the gram stain. Gram-positive bacteria have a thicker peptidoglycan wall and stain purple, whereas the more common gram-negative bacteria contain less peptidoglycan and do not retain the purple-colored dye. These gram-negative bacteria can be stained with a red counterstain and then appear dark pink. In the gram-positive bacteria, the peptidoglycan forms a thick, complex network around the outer surface of the cell. This network also contains lipotoic and teichoic acid, which protrudes from the cell wall. In the gram-negative bacteria, a thin layer of peptidoglycan is sandwiched between the plasma membranes and the second outer membrane. The outer membrane contains large molecules of lipopolysaccharide, and those are lipids with polysaccharide chains attached. The outer membrane layer makes gram-negative bacteria resistant to many antibiotics that interfere with cell wall synthesis in gram-positive bacteria. In some bacteria and archaea, an additional protein or glycoprotein layer forms a rigid paracrystalline surface called an S layer outside of the peptidoglycan or outer membrane layers of gram-positive and gram-negative bacteria, respectively. Among the archaea, the S layer is the most universal and can be found outside of a pseudomurin layer, or in contrast to the bacteria, may be the only rigid layer surrounding the cell. The functions of S layers are diverse and variable. They can attach to the cell to a surface or afford protection to the cell. They can even trap molecules close to the cell surface or act as a selectively permeable structure. You're seeing before you gram-positive, and on the left, you see the plasma membrane. You see the cell wall with that thick peptidoglycan layer. And on the right, you're seeing gram-negative bacteria with the plasma membrane, that thin layer of peptidoglycan, and then the outer membrane that encapsulates or closes in on that, and that's called the lipopolysaccharide layer. In some bacteria, an additional gelatinous layer, the capsule, surrounds the outer walls and can be detected relatively easily by staining and microscopy. A more loosely organized form of capsule that is harder to detect is called a slime layer. A capsule enables a prokaryotic cell to adhere to surfaces and other cells, and most important, to evade an immune response by interfering with the recognition by phagocytic cells. Therefore, a capsule often contributes to the ability of bacteria to cause disease. Some prokaryotes are motile. Many prokaryotic cells possess one flagellum at one end. Some possess one at one end of the cell, and some possess many flagella in clusters or distributed around the cell. While appearing similar, these structures evolved independently in bacteria and archaea. The central filament of bacterial flagella can be from 0.3 to 12 micrometers long and 10 to 20 nanometers wide, and is made from a single protein called flagellin. The analogous structure in archaea is called archaeolum. It has a central filament composed of different proteins related to the proteins found in the bacterial pili. The structures anchoring flagella to the membrane and cell wall are also different. Cell motility is due to rotation of the protein fiber, but this is powered by different energy sources, a proton gradient in bacteria and ATP in archaea. Pili are numerous fine structures that occur on cells of some gram-negative prokaryotes. They are shorter than prokaryotic flagella and only about 7.5 to 10 nanometers thick. Pili can be used to attach a cell to a surface and may be important in allowing colonization of tissues by pathogens. Some pili are adapted to be involved in the sharing of genetic information, and other specialized pili may also be used to cause the cell to twitch, which can result in directional movement. This is showing you that flagellar motor of that gram-negative bacterium. Some prokaryotes are able to form endospores. A thick wall develops around their genome, and a small amount of cytoplasm when they are exposed to environmental stress. These endospores are highly resistant to environmental stress, such as heat, and when environmental conditions improve, they can germinate and return to normal cell division and form vegetative cells after decades or even centuries of dormancy. Yes, centuries. The bacteria that causes tetanus, botulism, and anthrax are all capable of forming spores. With a puncture wound, tetanus endospores may be driven deep into the skin, where conditions are favorable for them to germinate and cause disease or even death. Relative to eukaryotic cells, prokaryotes have a relatively simple interior organization. They lack the extensive functional compartmentalization seen within eukaryotic cells, but they do have the following structures. Membrane folds. They have the nucleoid region. They have ribosomes, and prokaryotic ribosomes are smaller than those of eukaryotes, and they differ in protein and RNA content. Antibiotics such as tetracycline and chloramphenicol can tell the difference. However, they bind to prokaryotic ribosomes to block protein synthesis, but they do not bind to eukaryotic ribosomes. And they also have internal compartments. And I suppose one thing that I'll mention a bit more about here would be the nucleoid region. So prokaryotic cells do not package their chromosomes in a membrane-bound nucleus. Instead, their circular DNA chromosomes are condensed to form a visible region of the cell known as the nucleoid region. Many prokaryotic cells also possess plasmids, which are small extrachromosomal, independently replicating circles of DNA. Plasmids contain only a few genes, and although these genes may confer selective advantage, they are not essential for the cell's survival, as earlier mentioned. In sexual reproducing populations, traits can only be transferred vertically from parent to child. Prokaryotes do not reproduce sexually. However, they can exchange DNA between different cells of the same species and, in many cases, between cells of different species. This horizontal gene transfer occurs when the genes move from one cell to another by conjugation, requiring cell-to-cell contact, or by means of viruses, transduction. And then some species of bacteria can also pick up genetic material directly from the environment, transformation. All of these processes have been observed in archaea, but the study of archaeogenetics is hampered by the difficulty of culturing most species. We concentrate here on bacterial processes and primarily those of E. coli, which has been studied extensively. Natural transformation occurs in some gram-negative species and some gram-positive species, which are said to be competent. Transformation occurs when one bacterial cell has died or ruptured, spilling its fragmented chromosomal DNA or plasmids into the environment. This DNA can be absorbed by another cell and incorporated into its genome, thereby transforming it. The cell taking up the DNA is said to be transformed because it likely obtains new characteristics after transformation. Some species of gram-positive and gram-negative bacteria are competent for transformation, although the mechanisms seem to differ between the groups. The proteins involved in the process of transformation are encoded by the bacterial chromosome. The implication is that natural transformation may be the only one of the mechanisms of DNA exchange that evolved as part of the normal cellular machinery. The transfer of chromosomal DNA by either conjugation or transduction, which we discuss next, can be thought of as accidents of plasmid or phage biology, respectively. Transformation is also important in molecular cloning, which relies heavily on the bacterium E. coli. However, E. coli is not naturally competent. When transformation is done in the laboratory, it is called artificial transformation. Artificial transformation is useful for cloning and DNA manipulation. Horizontal gene transfer can also be mediated by viruses that infect prokaryotes. In generalized transduction, virtually any gene can be transferred between cells. In specialized transduction, only specific genes can be transferred. With generalized transduction, it occurs via accidents by way of the lytic cycle. So viruses package bacterial DNA and transfer it in a subsequent infection. Whereas in specialized transduction, it occurs via accidents by way of that lysogenic cycle. So it's an imprecise excision of the prophage DNA. So these phage carry both the phage genes and chromosomal genes. Lastly, we'll get to conjugation. Plasmids encodes functions that are not necessary to the organism, but provide a selective advantage in particular environments. For example, antibiotic resistance is not necessary, but it obviously is advantageous in the presence of the antibiotic. Some plasmids can be transferred from one cell to another via conjugation. The best known transmissible plasmid is the E. coli F plasmid, the fertility factor. Cells containing F plasmids are termed F positive or donor cells, and cells that lack the F plasmids are F negative or recipient cells. The F plasmid is an extra chromosomal piece of DNA that uses cellular machinery for replication. So the F positive cell produces the F pilus that connects to the F negative cell. Transfer of F plasmid occurs through conjugation bridge, and then the F plasmid is copied through rolling circle replication. The end result is 2 F positive cells. Briefly, F plasmid can be integrated into the bacterial chromosome, so events are similar to crossing over in eukaryotes, and it's such as in homologous recombination. So there's a high frequency of recombination, or the HFR cell. So the F plasmid integrated into the chromosome and replicated every time the host divides. And then the F plasmid can also excite itself by reversing the integration process. An accurate excision may offer picking up some chromosomal DNA by way of the F plasmid. During conjugation, the high-frequency recombination strains, the gene transfer of genes is linear and progressive. So genes farther from the origin of transfer will be transferred later, and then different marker genes appear in the recipient cell at specific times. So gene order can be mapped based on entry time. Some conjugative plasmids can require antibiotic resistance genes, becoming resistance plasmids, or R plasmids. The rapid transfer of newly acquired antibiotic resistant genes by plasmids has been an important factor in the appearance of resistant strains of pathogenic bacteria. In some cases, bacteria can become resistant to multiple antibiotics. Resistant plasmids often require antibiotic resistance genes through transposable elements. These elements can move from chromosome to chromosome, or from plasmid to chromosome and back, and they transfer antibiotic resistance genes in the process. If a conjugative plasmid acquires these genes, then the bacterium carrying it has a selective advantage in the presence of antibiotics. Just as in any organism, mutations can arise spontaneously in bacteria. Certain factors, especially those that damage DNA, such as radiation, ultraviolet light, and various chemicals, can increase the likelihood of mutation. A typical bacterium, such as E. coli, contains about 5,000 genes. The probability that mutation will occur by chance is about 1 out of every million copies of a gene. With 5,000 genes in a bacterium, approximately 1 out of every 200 bacteria will have a mutation. With adequate food and nutrition, a population of E. coli can double in 20 minutes. Because bacteria multiply so rapidly, mutations can spread rapidly in a population and can change characteristics of that population in a relatively short time. The ability of prokaryotes to rapidly adapt to selective pressures, such as in the presence of antibiotic in the body, can have adverse effects on human health. A number of antibiotic-resistant strains of bacterium, Staphylococcus aureus, termed methicillin-resistant Staphylococcus aureus, or MRSA, have been known in hospital settings for some time. Of particular concern among these strains is vancomycin-resistant Staphylococcus aureus, or VRSA. This appears to have arisen rapidly by mutation and is alarming because vancomycin is a drug of last resort in treating MRSA, which makes these strains and infections they cause very difficult to control. Staphylococcus infections provide an excellent example of the way in which mutations in the presence of strong selective pressures can bring about rapid change in bacterial populations. So CRISPR systems provide adaptive strains of prokaryotic genomes revealed repeated sequences with spacer regions called CRISPR. So those are clusters, regularly interspersed short palindromic repeats. So recently, these have been identified as a form of adaptive protection against viral infection. In response to viral challenge, bacteria and archaea will integrate short segments of viral nucleic acid in these CRISPR loci, then use them to produce an RNA that can be used to guide a complex that degrades viral nucleic acid. Much like restriction endonucleases, the CRISPR system has provided useful for molecular biologists for genome editing. A 2012 survey found CRISPR loci in 48% of bacterial genomes and 95% of archaeal genomes sequenced. If you consider how viruses outnumber prokaryotic cells in virtually every environmental context, this represents very strong selective pressure for a system that can remember previous infection to protect against future infections. The variation seen in prokaryotes manifests itself most strikingly in biochemical rather than morphological ways. Not only do they have great metabolic diversity, but they also exhibit considerable metabolic flexibility, which is not seen in any other group of organisms. To meet the growth demands, organisms require carbon for building, a source of electrons to use in redox chemistry, and energy for anabolic processes. The nutritional strategy of an organism is simply the ways in which an organism acquires carbon, acquires electrons for redox chemistry, and performs energy transformation. Fortunately, there are only two ways in which carbon, electrons, and energy can be obtained. Carbon is obtained in reduced forms by heterotrophs and in oxidized forms, CO2, by autotrophs. Chemotrophs transfer energy by oxidizing reduced chemicals obtained from the environment. Phototrophs, on the other hand, transform energy by harvesting light. Lithotrophs obtain electrons from reduced inorganic substances in the environment, while organotrophs obtain electrons from reduced carbon sources. Prokaryotes can be classified into 5 different nutritional types. However, there are times when a particular organism may switch nutritional strategy based on nutritional needs and based on the nutrients available in the environment. This rarely, if ever, happens with eukaryotes. Now that when we name a prokaryote nutritional strategy, we string together into one word the terms that describe the approaches to obtaining carbon, electrons, and energy. The 5 types are as follows. Chemoorganoheterotrophs. These organisms use reduced carbon made by producer organisms as sources of carbon, electrons, and energy. They contribute to the biogeochemical cycling of carbon. Nearly all pathogens use this nutritional strategy when they cause disease. The most common sources of reduced carbon in the environment of these organisms are lipids and carbohydrates. All animals are chemoorganoheterotrophs. Chemolithoheterotrophs. As with chemoorganoheterotrophs, the source of energy and electrons for this nutritional strategy is reduced chemical in the environment. However, the chemical is inorganic, not organic. The Greek word litho means stone. So these organisms are colloquially known as stone eaters due to the mineral nature of the source of electrons and energy. So they have a colloquial name, stone eaters. The source of carbon used by these organisms is a reduced form obtained from the environment. Only prokaryotes utilize this nutritional strategy. They play critical roles in the biogeochemical cycling of many nutrients. Chemolithoautotrophs. The source of carbon in these organisms is the oxidized atmospheric form of CO2. CO2 is reduced to usable organic forms using electrons obtained from reduced inorganic compounds. The oxidation of reduced inorganic compounds also provides the energy required to produce reduced carbon for biosynthesis. Given the use of reduced inorganic nutrients, such as ammonia and sulfur, the use of this nutritional strategy contributes greatly to the nutrient cycling in the environment. Photolithoautotrophs. This nutritional strategy also produces atmospheric CO2 to produce molecules for biosynthesis. The sources of electrons for such reductions are inorganic. You are likely familiar with green plant photosynthesis, which uses water as a source of electrons and produces NADPH to drive the Calvin cycle. Some prokaryotes use water as an electron source. However, others use hydrogen and different forms of sulfur as electron sources. Prokaryotic photosynthesis can differ significantly from green plant photosynthesis. Photoorganoheterotrophs. This is a particularly unusual nutritional strategy used by prokaryotes, such as the purple and green non-sulfur bacteria. The source of energy is light in this strategy. However, organic carbon is used as a source of carbon and of electrons. Regardless of nutritional strategy used, an energy transformation must ultimately convert one source of chemical potential energy into the form of chemical potential energy usable by all living organisms, ATP. The transformation of energy in one chemical potential energy source to a usable source for the cell involves either photosynthesis, respiration, or fermentation. There are significant differences in how prokaryotes and eukaryotes perform these processes. In eukaryotes, cellular respiration uses an electron transport chain to recycle electron donors into electron acceptors, reduce oxygen to water, and produce a proton motive force to drive ATP synthesis. The final electron acceptor is oxygen. So these respirations are aerobic. Prokaryotic respirations, on the other hand, show remarkable diversity of both electron donors and electron acceptors. Respirations that use a final electron acceptor other than oxygen are anaerobic respirations. Such acceptors include oxidized substances such as nitrate and sulfate. Some bacteria can even use carbon dioxide as an electron acceptor in respiration, producing methane as gas as a product. Other bacteria are known to reduce organic compounds in the electron transport chains. And briefly, fermentation may be used in a cell when a terminal electron acceptor is not available to allow respiration. Fermentation can recycle reduced electron acceptors such as NADH to form their oxidized form, which is needed in oxidation reactions to drive the formation of small amounts of ATP. There is some diversity in the kinds of fermentations seen in eukaryotes. Yeasts can ferment sugars to produce ethanol, animal muscle can ferment sugars to produce lactic acid, and under certain conditions, plant cells can ferment sugars to produce ethanol. Prokaryotes, however, show much greater diversity of fermentations. A variety of metabolic pathways used by prokaryotes can produce different alcohols and acids from the fermentation of pyruvate, the product of glycolysis. Some of these products have industrial or commercial applications and contribute to the flavorings of milk and cheese products. The production of many people's favorite indulgence, chocolate, is possibly only due to the formation of cocoa. Thanks a lot, bacteria. And perhaps the most unusual fermentation seen in prokaryotes is performed by the Clostridium genus of bacteria. This genus includes species that are industrially valuable to production of certain fermentation products, but also includes dangerous human pathogens. Clostridium tetani is a causative agent of tetanus and is introduced into the body most commonly through deep puncture wounds to the skin. The environment deep in the wound is anaerobic, and Clostridium tetani can grow by fermenting the abundant amino acids in tissues. Clostridium botulinum is well known as the pathogenic bacteria producing Botox toxin used in cosmetic enhancements to reduce wrinkling of skin. This bacteria can be introduced into foods during the canning process, and if an abundant source of protein is available in the anaerobic interior of the can, the bacteria can ferment the constituent amino acids to support growth. Ingesting the toxin produced by the pathogen along with the food can lead to serious illness or even death. This pathogen is particularly dangerous to young children with poorly developed gastrointestinal flora. Prokaryotes were largely responsible for creating the current properties of the atmosphere and the soil through billions of years of their activity. Today, they still affect the earth and human life in many important ways. Earth is critically dependent on the cycling of chemical elements between organisms and the physical environments in which they live, that is, between the living and non-living elements of ecosystems. Nutrient cycling involves two fundamental processes: decomposition, in which nutrients are released to the environment from dead organisms, and fixation, in which oxidized nutrients are made available for use by heterotrophs. Prokaryotes, algae, and fungi play many key roles in this chemical cycling, a process that you'll learn more about if you take Principles of Biology 2. Decomposition. The carbon, nitrogen, phosphorus, sulfur, and other elements of the biological systems come from the physical environment, and when organisms die and decay, these elements return here. These elements return there. Certain prokaryotes and fungi are decomposers. responsible for the majority of nutrient cycling in ecosystems. Decomposers carry out the decomposition stages of biogeochemical cycles, which release matter from decaying organisms, freeing those nutrients for reuse in the environment. Fixation. Other prokaryotes play important roles in the fixation, helping to return nutrients from oxidized inorganic forms to reduced organic forms that heterotrophic organisms can use. The role of photosynthetic prokaryotes in fixing carbon is critical to the biogeochemical cycling of carbon. The carbon compounds that plants, algae, and photosynthetic prokaryotes produce from CO2 pass up through the food chains to form the bodies of all the ecosystem's heterotrophs. Some prokaryotes, called methanogens, can also contribute to the reduced carbon in the environment by producing methane. Methanotropes, on the other hand, can oxidize methane to CO2. Nitrogen. Less obvious, but no less critical to life, is the role of prokaryotes in the cycling of nitrogen. The nitrogen in the Earth's atmosphere is in the form of nitrogen gas. The triple covalent bond that links the two nitrogen atoms is not easy to break. Among Earth's organisms, only a very few species of prokaryotes are able to accomplish this, reducing nitrogen gas, or N2, to ammonia, N3, which is used to build amino acids and other nitrogen-containing biological molecules. When the organisms that contain these molecules die, the decomposers return nitrogen to the soils as ammonia. This is then converted into nitrate by nitrifying bacteria, making nitrogen available for plants. The nitrate can also be converted back into molecular nitrogen by denitrifiers that return nitrogen to the atmosphere, completing the cycle. To fix atmospheric nitrogen, prokaryotes employ an enzyme complex called nitrogenase, encoded in a set of genes called NIF, nitrogen fixation genes. NIF genes, or nitrogen fixation genes. The nitrogenase complex is extremely sensitive to oxygen and is found in a wide range of free-living prokaryotes. Like all organisms, prokaryotes need places to live that supply their nutritional demands, and where they can invade or deter predators. Some homes may be found in the environment, but can also be on or in other organisms. Prokaryotes can exist in a number of types of relationships with organisms in all the kingdoms of life. An ecologically important example of a mutually beneficial association between bacteria and plants is the relationship between nitrogen-fixing bacteria, primarily in the family Rhizobiaceae, and leguminous plants, such as peas and soybeans. These bacteria can establish infections in the roots of leguminous plants, where they produce signaling molecules that induce the root to produce a nodule where the bacteria take up residence. Shown here, root nodules. The bacteria are capable of performing nitrogen fixation to produce reduced nitrogen, primarily ammonia, which can be metabolized into organic forms of nitrogen such as amino acids. The bacteria receive photosynthetic metabolites from the plant while providing fixed nitrogen in the form of amino acids. This relationship allows leguminous plants to flourish in the soil depleted of reduced nitrogen and can reduce the amount of fertilizer used agriculturally. Excess fertilizer can lead to increased bioavailable nitrogen and phosphorus in agricultural runoff. Lastly, not all plant prokaryote interactions are mutually beneficial, however. Another member of the Rhizobiaceae, Agrobacterium tumefaciens, can establish a parasitic relationship with numerous plants, including important crop species. So that what results is called crown gall disease, causing the tumor shown, here. The inability of some prokaryotes to cause disease, the exploitation of mammals by prokaryotes, has been most extensively studied. In addition to the disease-causing relationships we explore later on, there are many beneficial relationships between mammals and prokaryotes. Prokaryotes and digestion in ruminants. Prokaryotes and digestion in ruminants. Mammals have evolved diverse structures to facilitate the digestion of diverse foods. Some herbivores have digestive tracts that allow ingested foods to be held long enough that anaerobic bacteria can digest cellulose. The main macromolecule in plant matter. This allows the extraction of essential calories and nutrients from plant material. In ruminants, a specialized digestive organ called the rumen holds cellulose-rich material that can be digested by bacteria. The anaerobic rumen bacteria hydrolyze cellulose to glucose, which can be metabolized to make a variety of fatty acids. These can then be absorbed and used as a source of energy by carbon by the animal. Unfortunately, for these helpful bacteria, many are digested farther down the GI tract, hoping to meet the other nutritional needs of the animal. And the human microbiome and its role in health. Humans are excellent hosts of bacteria. Although some of those bacteria may be harmful, the overall majority are beneficial, and many play a role in protecting us against pathogens. Humans can host bacteria on their skin and in the respiratory, urogenital, and gastrointestinal tracts. The collective population of bacteria living on or in a human is called the human microbiome. Scientists are concerned primarily with the gut microbiome and whether differences between individuals contribute to differences in health or predisposition for certain conditions. The use of organisms to remove pollutants from water, air, and soil is called bioremediation. The normal functioning of sewage treatment plants depends on the activity of microorganisms. In sewage treatment plants, the solid matter from raw sewage is broken down by bacteria and archaea naturally present in the sewage. The end product is methane gas and is often used as an energy source to heat the treatment plant. Biostimulation adds nutrients to encourage the growth of naturally occurring microbes. And this has been used to encourage the growth of naturally occurring microbes that can degrade crude oil spills. This approach was used to clean up some 3 million barrels of oil that spilled down in the Gulf of Mexico in 2010 from the Deepwater Horizon oil rigs when it exploded. And it caused the largest ever marine oil spill with a devastating effect on marine life. Similarly, biostimulation has been used to encourage the growth of naturally occurring microbial flora in contaminated groundwater. Current efforts include those concentrated on the use of endogenous microbes such as Geobacter to eliminate radioactive uranium from the groundwater contaminated during the Cold War. Human bacterial disease. So this is where we'll end the chapter. So in the early 20th century, before the discovery and widespread use of antibiotics, infectious diseases killed nearly 20% of all United States children before they reached the age of 5. Sanitation and antibiotics considerably improved the situation. In recent years, however, we have seen the appearance or reappearance of many bacterial diseases, including cholera, leprosy, tetanus, bacterial pneumonia, whooping cough, diphtheria, and Lyme disease. Members of the genus Streptococcus are associated with scarlet fever, rheumatic fever, pneumonia, necrotizing fasciitis, and other infections. Tuberculosis, another bacterial disease, is still a leading cause of death in humans worldwide. We know now that the bacterium H. pylori can cause both gastric ulcers and cancers associated with gastric lymphoid tissue. Bacteria have many different methods of spreading through a susceptible population. Tuberculosis and many other bacterial diseases of the respiratory tract are mostly spread through the air in droplets of mucus or saliva. Diseases such as typhoid fever, paratyphoid fever, and bacillary dysentery are spread by fecal contamination of food or water. The pathogens that cause Lyme disease and Rocky Mountain spotted fever are spread to humans by arthropod vectors such as ticks. Bacteria cause disease in a variety of ways. To cause disease in humans, bacteria must establish an infection, evade the immune system in order to proliferate, and then either cause direct tissue damage, release toxins to harm, or evoke an inflammatory response that harms the host. All pathogenic bacteria have specific structures or physiological processes that allow them to use human hosts as environmental niches where they can inadvertently cause disease or even death. The first step in infection is the establishment of the pathogen in the host. The initial site of infection is often where a mucosal surface is exposed to the environment. It is helpful to keep in mind that the human urogenital systems are open systems, which allow organisms access to mucosal epithelia that are exposed to the environment. This is also true of the human respiratory and gastrointestinal systems. Initially, attachment involves specific and non-specific adherence of the pathogen to the tissue. Attachment is promoted by valence factors, which are specific genetically determined features of a pathogen. These include pili, capsules, S layers, and cell wall components such as the lipopolysaccharide and teichoic acids. Immune invasion by pathogens. It is not of interest of pathogen to kill the host, since the host is a source of nutrition and protection. Therefore, pathogens need strategies to replicate without being eliminated by the host immune system and without killing the host. Disease and death are unfortunate side effects of infection rather than a strategy of most pathogens. To establish relatively long-term infections that allow replication of the pathogen, a host immune system must be evaded, and again, specific valence factors facilitate this invasion. Not all pathogens cause disease at the point of infection. Some can produce secreted proteins that break down host tissues to allow the pathogen to spread in the body. These proteins are collectively called invasins that can be a major cause of disease due to tissue damage. Invasins usually cause localized tissue damage to facilitate pathogen spread by degrading molecules such as hyaluronic acid and collagen in connective tissues. Toxins produced by some pathogens may act locally to cause tissue damage. However, some can cause damage distant from the localized site of pathogen growth. This is often the distinction made between invasins and toxins. So important bacterial diseases include anthrax, caused thereby Bacillus anthracis, botulism, chlamydia, cholera, dental caries from the genus Streptococcus, diphtheria, gonorrhea, and Hansen's disease, also mentioned earlier as leprosy. There is also Lyme disease, peptic ulcers from Helicobacter pylori, which I mentioned earlier, from H. pylori, the plague, pneumonia, tuberculosis, typhoid fever, and typhus. So as just mentioned, this is how bacteria cause disease. Gaining entry, colonizing, evading the immune system, because the key is not to kill the host, spread to other sites in the body, and then causes the damage by the production of toxins or by triggering inflammatory responses. So tuberculosis, which I won't go through all of these, it's been a scourge for thousands of years. It afflicts the respiratory system. It is easily transferred from person to person through the air, and it is multi-drug resistant strains that are very alarming. So Helicobacter pylori causes ulcers and even cancer. So they can cause stomach ulcers, though they are treated by reducing stomach acid, and they can also cause gastritis, which is treated by antibiotics. Then there are sexually transmitted infections, and many of them are caused by bacteria. So it says a number of bacteria cause sexually transmitted infections, otherwise known as STIs, and three particularly important ones are gonorrhea, syphilis, and chlamydia. So gonorrhea is one of the most prevalent communicable diseases in North America. It is transmitted through exchange of bodily fluids and can pass from mom to baby via the birth canal. Chlamydia is that silent STI, and the incidence has skyrocketed, and it can cause pelvic inflammatory disease and even heart disease. And then there's syphilis. It is transmitted through sex or contact with an open canker. It can pass from mom to baby via the birth canal. And there are four distinct stages. The canker, which is highly infectious. The rash, it is also infectious. There is that latency period, which is no longer infectious but attacking internal organs. And then lastly, the damage now evident, causing heart disease, mental deficiency, and even nerve damage. So this is showing those trends in STIs, or at least sexually transmitted disease, in the United States. So although syphilis was down, syphilis rates are going back up. It looks to be that gonorrhea rates did increase, but look to be decreasing a bit. And chlamydia rates have skyrocketed and look to still be on the rise or even plateauing, but still, I say on the rise.